Hi, my name is Prateek Raikwar, and today I'll be talking about the LGA5 nucleus, dynorphin, and their translational applications. Okay, so previously I presented on our lab's discovery of the LGA5 nucleus brainstem neurons, which express dynorphin and have kind of a robust inhibitory projection to lamina 1 of the dorsal horn in the spinal cord. You can see that here in this figure on the right. As you can see with this data on the left, though, chemogenetic activation of these neurons suppresses itch sensation in mice through a top-down inhibitory mechanism that affects the spinal parabrachial tract. So what I'm doing now is trying to write a literature review about the general role of dynorphin in the suppression of itch. Okay, so the hypothesis of the review is that the kappa opioid system, which is the system dynorphin acts upon, is involved in the suppression of itch along the spinal parabrachial tract. So my study objectives then are to compile literature to show how dynorphin acts along this pathway, and that involves more deeply gathering data from studies that shows where dynorphin acts along the spinal parabrachial tract, as well as to what degree dynorphin acts to suppress the itch sensation at those sites. I also aim to analyze the field's data to suggest the most promising areas of future research for the kappa opioid system and itch. My methods involved, of course, first defining my hypothesis and research objectives, and then screening the field of research on PubMed. There was 139 preliminary articles representing search terms such as itch and dynorphin, among others, and I deemed 34 of those irrelevant because based on like title and abstract information. I went through the 105 remaining articles and deemed 22 of those irrelevant again, and now I'm writing the review with 83 papers as my current goal. So what have I found so far? As I mentioned before, here in orange, we have the spinal parabrachial tract, and it starts with the periphery with the dorsal root ganglion. It travels up the spinal cord, and it ends in the brain with the thalamus. So basically what I found is that dynorphin seems to suppress itch neural transmission at 3 major sites, one at the dorsal root ganglion site, one at the gastrin-releasing peptide receptor site with B5I neurons releasing dynorphin, and one at the neurokinin 1 receptor site with dynorphin released from LGA5. So because this review is based on basic biomedical research at this point and not any intervention with humans, I would say all this knowledge is at the T0 stage of the translational spectrum. I wouldn't say it's quite T0.5 because we haven't actually identified the B5I neurons nor the LGA5 neurons in humans yet, meaning we can't really translate this model to humans at all. So naturally, you'd think that the next best move to make this more translational would be to search for LGA5 and B5I neurons in human tissues, but our lab has had some trouble with that. So I propose instead that we run a T.5 study with humans and drugs with a larger goal of seeing if it's even worth continuing our search for these neurons in humans. So how would that work? So basically, it would be like a mini clinical trial type of thing where the patients in our trial would be ones with chronic itch, and they would receive one of 5 treatments. The first is difelikefalin, which is a peripherally selective dynorphin agonist, meaning it'll only really act at these dorsal root ganglion receptors. Next is cyclosomatostatin, is an somatostatin antagonist, so it'll decrease the suppression of the somatostatin on B5I neurons, causing them to release more dynorphin at GRPR neurons. There will be a combination of those 2 treatments here. And then finally, there's going to be a nalfurafine treatment, which is a general dynorphin agonist, meaning it'll act as dynorphin at these 3 sites. Okay, so, and then there's going to be a placebo, of course, too. So we would monitor itch using observational and self-report data over a set time period to analyze how each treatment kind of affected itch response for these patients. So my hypothesis with this would be that there would be a summative effects in terms of dynorphin recruitment and suppressing the itch response. In other words, difelikefalin alone and cyclosomatostatin alone treatments would cause a lesser degree of itch suppression than their combination, which would cause a lesser degree of itch suppression than nalfurafine because there's a increasing amount of dynorphin recruitment. If this held true, this would yield knowledge to improve the translational potential of biomedical discovery of B5I and LGA5 neurons, meaning we would be more likely to find them in humans, and that ultimately goes to show that my study is one that is T0.5. These are my sources. Thank you.